Welcome to my talk. Today I'm going to talk about how to build an ecosystem model, specifically an end-to-end ecosystem model, what that entails, and what we did to build one for the Northern California current. And then at the end, I'm going to end by talking about integrating some of this information and how we might think about open science during this process. So salmon are an iconic species that are broadly distributed across the Northern Hemisphere. They have cultural significance to many groups. They're ecologically important as they cycle nutrients between the ocean and freshwater, and are economically important and support a number of major fisheries. Yet many salmon are declining globally. And it is thought that most of the mortality during a salmon's life cycle occurs in the ocean, which is the stage we know least about, and a warming ocean isn't helping their survival. Many populations of Chinook salmon, for example, are well studied in freshwater, but poorly understood in the ocean. And research is made much more difficult in the ocean because it's so vast and it's unforgiving to research gear, which together make ocean research prohibitively expensive. But a part of this gap also might be due to attitudes. You'll often hear people saying that we can't do anything out in the ocean. We just have no management actions out there. But is this really true? So keep this in mind, and I'll come back to it shortly. So many modern freshwater fishery management laws can be dated back to the 1200s in the Magna Carta, when King John made it illegal to block fish passage in some rivers. And there's a long history of Western freshwater fish management, starting in Europe and moving across the Atlantic to the east coast of North America before the Pacific. And historically, we've mostly managed our fisheries with individual species data and single species models. So how is a particular stock doing effectively in isolation? But recently, we've been collectively pushing towards ecosystem-based fisheries management. And we've started collecting a huge amount of scientific survey data on everything from phytoplankton, copepods and jellies to seabirds and whales and other marine mammals. So when we think about what we can do to understand what happens to this salmon in the ocean, we ought to realize that salmon don't exist in isolation. They live in a world with predators, competitors, and prey. Well, we catch those predators, competitors, and prey in our commercial and recreational fisheries, as well as the salmon themselves. So these are huge management levers that we have. Additionally, we protect some groups under the Marine Mammal Protection Act or the Migratory Bird Act. And on the other side of that coin, we also lethally remove pinnipeds from below Bonneville Dam and relocate nesting birds within the lower Columbia River. So there are management actions that we can do, but they're often incredibly costly and have a high degree of uncertainty associated with their success. In a terrestrial or freshwater system, we might be able to manipulate some of these things in smaller scale experiments. We can do some sort of predator exclusion or supplementing prey, for example. Yet this isn't so straightforward out in the open ocean, and it's nearly impossible to do on an ecosystem scale. So what we need is a model, something that can allow us to understand how all these pieces fit together, so we can inform whether or not any particular action might be worthwhile. So if we want to stop declining ocean survival for salmon, we need a model of the ocean ecosystem, one that will allow us to adjust hypothetical management actions. And this can allow us to assess what in the ecosystem is causing declines and what we might do about it. So much like you might build a structural model of a building or of a college campus or resort to make sure that all the pieces actually fit together the right way before getting boots on the ground and building the thing, we set out to build an ecosystem model that would allow us to understand more about how our local ocean ecosystem operates in a holistic sense. So partially pictured here is the Northern California current, which extends from Vancouver Island, British Columbia, Cape Mendocino, California. And this is where the juvenile salmonids coming out of the Columbia River first meet the ocean. And many of the wild stocks in this basin are threatened or endangered. And so this is the region that we focused on for our model. So what does it actually take to build one of these things? Well, first, we have to build a food web. So that is, you have to define linkages between predators and prey. So how much of a particular prey is a predator eating? Is it a little bit or is it a lot? And then once this is done for every possible linkage in the network, you have a food web or a diet matrix. Then one needs to parameterize the biomass values. So some groups might be very large, and these groups should be consuming more prey in the system than smaller groups. Makes sense, right? So this allows the total consumption by predators to be scaled by their relative biomass values in the ecosystem. And then lastly, if we want to accurately map consumption of one group on another for each group in the model, we have to define biomass-specific consumption rates of both predators and prey. So that's this Q over B term here. And sometimes you might see it C over B, which makes more sense for consumption. But in a second, we'll see the equation for the model, and it might make more sense why it's Q. And so you might think of this consumption rate as something like appetite. So some groups consume more for their body weight than others. We also have to define biomass-specific production rates. That's this P over B term. That is how much energy one consumes goes towards growth of the group in the model. And we also have to define losses due to metabolism and losses, which then becomes detritus and can be consumed by other groups in the model. So let's put all these pieces together a little bit more precisely. And I know not everyone likes to look at math, so we'll walk through this nice and slowly. But notice at the very end here, there is already a C term. And so I'll explain what that is in a second, but that's why sometimes we see this Q over B instead of C over B. So first, just broadly, everything on the left side of the equation is contributing to production or growth, if you want to think of it that way, of group I in the model. Everything on the right side is consuming or removing biomass or energy from group I. So starting on the left, we have biomass of the group of interest. So notice the subscript I there just means that there's a term here for every functional group in the model. We have the biomass specific production rate that I introduced earlier. So that's going to be multiplied by the biomass to get at what the growth in the next stage will be, the next step of the model, time step. We have an ecotrophic efficiency term that I won't get into here, but it's essentially, what proportion between 0 and 1 of the energy produced by the group I actually goes into the growth. So obviously this number can't exceed 1. That would be impossible. You can't produce more than you consume, but that's sort of for the mass balancing stage, and I won't get into it here. Then there's an immigration rate. So you can have biomass immigrating into your model. There's a biomass accumulation term. So perhaps a population of interest we know is increasing over time or decreasing over time. We can account for that here. And then we have the biomass of the predators. So starting with predator J, notice that's a different subscript. And we have the consumption rate of that predator J. And then that's all multiplied by the proportion of the diet that is that prey group I, right? And so all of the predators will be summed up for each group, because it's not just one predator that consumes each prey, right? There's many, many predators out there. And so those will all be summed up. And then there can be emigration out of the model. And then there's fisheries catch. So that's that C term. So now that we have the scaffold of our model built, we can see how the pieces fit together. And we want to see what happens to the entire structure if we start removing or changing pieces in the system. Will it collapse? What will happen? So what can we actually tweak here to understand this? So I'll make some changes here to the food web itself in the diagram, and then also the equation down below. So one thing we can do is we can alter a linkage. We can reduce the amount of predation that a particular predator or suite of predators has on a particular prey. We can also reduce the biomass of a particular group or suite of groups and assess the impact that that action would have on the entire ecosystem. We can alter fisheries landings in different ways. And since primary productivity in our model is linked to physical oceanography, we can let varying ocean conditions, which will propagate throughout the food web, to change. So this allows us to run oceanographic scenarios that mimic future climate projections so that we can maybe be better prepared for changes that will occur to the ecosystem in the future. Okay, so that's sort of the broad overview of how to build an ecosystem model or an end-to-end ecosystem model. And so now, how do we actually parameterize this model for our region of interest? What did we do? So let's start with these biomass terms, both the prey and the predator, which are actually the same, right? Because many predators are also prey to other things. And as I mentioned earlier, lots of scientific survey data are being collected on many different critters. And we were able to access and use many of these surveys. So the Northwest Fisheries Science Center at NOAA runs an acoustic trawl hake survey, where they pair acoustic data with trawls to estimate biomass of hake and krill. We use a juvenile salmon and ocean ecosystem survey to parameterize groups such as salmonids, seabirds, and jellies. And a rockfish recruitment survey allows us to parameterize many groups, including jellies again, juvenile fish, shrimp, and krill. The West Coast Groundfish Survey provides many bottom-dwelling fishes, such as adult rockfish, flatfish, and crabs. The Southwest Fisheries Science Center also runs an acoustic trawl survey, but for coastal pelagic species such as sardine, anchovy, and herring. And Oregon State University's Newport Hydrographic Line Survey provides many of the smaller things from phytoplankton and small invertebrate larvae to krill species information. So now we've got that on here, showing you where those biomass values come from very, very broadly. What about the diets? Where do we get those? So to build our diet matrix, we relied on many sources of literature and individual studies that are, some of them are unpublished. And importantly, we relied on a Southwest Fisheries Science Center diet database that incorporates a bunch of the NOAA data sets that are available and just sort of puts them at one central location. So that was super helpful, highly collaborative effort here to get all these diets. And then lastly, the last one that I'll talk about now is fisheries catch. So where do all these data come from? So the Pacific States Marine Fisheries Commission has a couple of portals. The ACFIN is the commercial fisheries information, and RecFin is the recreational fisheries information network. And this is a huge collaborative effort across state agencies and some federal agencies as well. And so, you know, the last two things I'll mention, the Q over B and P over B terms, the consumption and production rates, those are all from literature sources that are, you know, that are not necessarily publicly available. They're all sometimes behind paywalls and things like that. But the point here is this is a highly collaborative effort. And so all these different data sets are used to parameterize this model, including from the literature. And I hope it's evident that it's important that we make all these data available to the world, right? Because I'm not the only one building these types of models. People have ecosystem models for ecosystems all over the world. And just within the Northern California current, there are at least 2 other overlapping models which require just as many sources of data. So if we're hoping to go the route of ecosystem-based fisheries management, and if we think those data collection efforts are important, we need to make those data available so that they can be used for all kinds of modeling efforts so we can better understand the world around us. And this call to share data has also come from the top down as well. So in 2013, the White House signed an executive order instructing all federal agencies conducting research to create a plan for making all research results publicly available. Then a couple of years later, NOAA came out with their plan to increase public access to research results, or PAR. And each year, the NOAA's PAR compliance rate is growing. It's increasing each year. But we're still far behind other government agencies. And so this might be a sign that fisheries in general is behind in open science practices and sharing their data with everyone else. And there are many others pushing for more open and transparent natural sciences. So this group called SORTI started within the last couple of years and is essentially an organization that is committed to helping the scientific community adopt open science mindsets that will surely speed up the progress of understanding the world around us. So one relevant example is how quickly we were able to collectively understand and respond to COVID-19 because of open science practices. Imagine if we could have that kind of coordinated and collaborative response to declining populations of wild salmon. With this said, I understand that many have hesitations in sharing their data with the world. There are many reasons. So a few of us from this SORTI group got together and put this document in a preprint server outlining the top reasons we think researchers don't want to share data in code. And we highlight some counter arguments there. So I really encourage you to check that out if you're in the camp of thinking open data is a bad idea. And I also encourage you to just contact me directly and share your reasons why you don't want to share data or the sort of pitfalls you've come across if you need any help figuring out how you might share your data more effectively. Because ultimately, whether our goal is to save iconic species from extinction or to better manage our fisheries, we all benefit from sharing information with each other. Because the more information we can incorporate into modeling efforts and understanding of the world around us, the more informed we're going to be in decision-making processes. And resisting open science practices doesn't just hurt your career through lower citation rates and things like that, but it also hurts the entire field of fisheries biology. So with that, I have a ton of people to thank. Jim Ruzicka and Lisa Crozier have been my mentors through this entire project. So I really appreciate all of their time and energy. And then a bunch of people have contributed data and expertise in this ocean ecosystem. So with that, I'll end and thank you very much for listening.